welcome to our May webinar. This is with Dr. Tom Robinson. So we're very, very pleased to have you with us, Tom. I'm going to it's just great to have you here. Tom's a lecturer at the Healthcare Technologies Institute at the University of Birmingham. And I think I should be clear, Birmingham, UK. I think there's a Birmingham in the States as well.
It's Birmingham, Alabama, yeah.
There is one, yes, there's one. So, so welcome everyone to the webinar. As usual, we have, can I advance this? Just a bit of housekeeping at the beginning. It's a webinar, so I don't think attendees will be able to put their cameras or microphones on anyway. Please do use the chat. For any questions, just add them in as we go along, and we'll ask questions of Tom at the end. The webinar will be recorded and it will go up on our website as with the previous ones. Please don't take screenshots or film it yourself separately. It will be made available publicly on the website. We've got the Q&A as well. You can use the Q&A or the chat. And just in terms of confidentiality, again, don't take your screenshots and share them around. We will publish the recording of the webinar in a week or so's time. What we got here? I'll just quickly plug the next month's webinar, which is our June webinar with Dr. Mark Popenhagen, but everyone will get a reminder about that going forwards. I will stop share and just say thank you again to Tom for being here with us. As I said, Assistant Professor of Healthcare Technologies and a lecturer at the University of Birmingham, working on the chemistry of long sugar molecules called polysaccharides and how they can be used in healthcare products. I'm going to shut off my microphone, shut off my camera, and over to you, Dr. Tom Robinson. Thank you.
Thank you very much. Thank you so much for having me at this series, and thank you all for coming and giving me some of your time. So maybe just to introduce myself a little more, especially in the context of questions. I don't know who's in the room, but I imagine a lot of you know a lot more about EB than I do. So I'm very much coming at this from a material science background and trying to apply what we know to see how we can help people with EB. So today what I want to talk about briefly are some of the oral conditions that can happen with people with EB, particularly scarring. And this is most prevalent in RDEB, but other subtypes as well. And the work we're doing with polysaccharides, which I'll introduce as well a bit later, and how we can kind of, well, what we can do, how we can try and help these people. So just talk a little bit about the epithelium. So the epithelium is essentially the skin, but also the inside of the mouth and essentially all the internal linings of the body. And it's a stacked layer of multiple different types of cells. And as I'm sure a lot of you know a lot better than I do, all types of EB are characterized by fragility in this tissue. So different subtypes have this fragility occurring in different areas. But essentially what it means is that the skin, different layers can become detached from each other. And again, there's lots of different biologically things on the side that cause that. So in the mouth, especially, what that means is that everyday activities, things like eating, chewing, brushing the teeth, causes the epithelium on the inside of the mouth to break down a little bit. And that cause can result in blisters, it can result in ulceration, and then that often results in scarring. And as the condition goes on, this repeats, the scarring is progressive. So you get more and more buildup of scar tissue in the mouth, which then becomes a vicious circle where these same activities that I think many people without EB take for granted become so much more difficult: chewing, swallowing, maintaining a brushing routine, speaking. As this progresses even further, you can get the tongue essentially tethered to the floor of the mouth through scar tissue. The opening of the mouth becomes smaller. The amount you can open your mouth just because of all this scar tissue buildup. So to take a step back, I want to really talk about what scarring is. And I'm going to use the analogy of this brick wall. So you can imagine the epithelium as being a bit like a brick wall where the bricks are the cells. So you've got lots of different cell types. As we saw in the previous slide, these are actually differentiated down. So actually this wall is several layers thick, but if we imagine it as a single layer, and it very much has the function of a wall. So the skin, the epithelium really is there to act as that primary barrier against the outside world and the rest of your body. So the bricks are the cells, and then the cement in between the bricks is what we call the extracellular matrix. And that's really what binds the cells together. It's where the cells live. And it's a bit of a back and forth. So the cells take cues from the environment, and they also lay down the environment in which they live. So what happens when we have an injury in the epithelium? How does the body respond? Well, the short answer is it's incredibly complicated, and I'm by no means a biologist, but essentially there's a couple of phases. So inflammation, the body will send a lot of cues, chemical and biological cues, to that site of injury to try and resolve the issue as quickly as possible. So things like platelets, you can imagine if you have bleeding, the first thing the body will try and do is cause clotting, stop that bleeding from continuing. It will then send other factors into the area, one of which we'll focus on in a second. It will then cause the cells in that area. So fibroblasts are the key cells that I'm going to talk about today, but there are other cell types in there, keratinocytes, other endothelial cells, like kind of blood vessel type cells. And then you get gradual remodeling, or we would hope that you get gradual remodeling. The three things that I want to talk about specifically are transforming growth factor beta 1. So this is one of the signals that your body sends to an area of damage in the epithelium to try and resolve it. Collagen is the main thing that cement in between the cells is made of. So again, that gets massively chucked out as well to try and close that gap. And the cells that do that are called myofibroblasts. So what a myofibroblast is if you take a fibroblast that's perfectly happy in the epithelium, when exposed to damage or other factors, transforming growth factor beta 1, which is one of those key signals I mentioned, comes in, attaches to the receptor on that fibroblast, a load of intracellular signaling happens, and what it does is it changes that cell into an activated fibroblast or a myofibroblast. So what that myofibroblast cell does, it's a bit like a fibroblast, but 10 times as active. So one of the key distinguishing factors of a myofibroblast is it has these fibrils that are made of something called alpha smooth muscle actin. What that does is it gives these cells contractile ability. So it allows them to pull tissue together. So myofibroblasts are very contractile. The other thing they do is that they can churn out a lot of collagen, so a lot of that cement material in a very, very short space of time. And I think it's worth saying at this point that this is actually incredibly important in the healing process. So because the skin is that barrier function and it stops everything coming from the outside world into your body, it's very important for the body to try and heal that or close that as soon as possible. What scarring is when it does that suboptimally. So going back to our brick wall analogy, you can imagine these myofibroblasts as being these big super bricks. And what the big super bricks will do is they will use that contractile ability to close that wound up, and they will lay down extra collagen, extra cement, and often that will result in a small scar. Because it's laying down this collagen incredibly quickly, it can't really pay attention to how this collagen in your skin is normally laid down. It just churns it out as quick as possible, and that's what results in scarring formation. The problem we have is when these activated myofibroblasts don't go away. So normally once you've laid down that tissue, once the wound has resolved itself, those myofibroblasts regulate themselves and they disappear, they die. If they don't and they stick around, they will continue laying down more and more matrix, more and more collagen, and that is essentially what a scar is. It's too much collagen having been laid down too quick. So what can we do? So this is essentially what's happening in the mouth of a lot of people with EB. How can we solve this? Well, there's a couple of different ways we can try and get around this. So the first one, perhaps the most effective, is just to stop the injury cascade altogether. So if we're talking about the mouth of people with EB, if you can stop those frictional forces from causing damage to the epithelium, everything else becomes moot. You've stopped it at the cause. Obviously, that's easier said than done, but that's one area we can look at. The second one is preventing those biological cues. So that little diagram I showed where the TGF-beta sticks onto a fibroblast and then causes it to turn into a myofibroblast. Maybe we can try and interfere with that pathway somewhere. And then the third one at the far end is maybe once we even, if we're at the point where we have all these extra myofibroblasts, can we prevent the physical formation of scar tissue? Can we stop that collagen, that cement from being churned out as quickly as it is and causing all this scarring that's causing us so many issues? So I'm now going to take a bit of a turn. I'm going to talk about polysaccharides. So these are the molecules that we're going to be interested in. The reason I'm interested in them, as I'll come into a bit, is they're used in all sorts of food and pharmaceutical formulations. But essentially what saccharide means sugar. So, and poly means many, and there's lots of different saccharides. So hopefully a lot of you will be familiar with glucose. So that is the main fuel that the body uses for energy at the end of the day. So sports drinks like Lucozade have lots of glucose in them because your body can use it incredibly quickly. And that's a monosaccharide. It's just this single ring. That's what it is. Your body can break that down. Table sugar is a disaccharide. So that is a glucose stuck to a fructose, and that makes sucrose, which is what table sugar is. Again, quite small. Your body can use that very readily. Polysaccharides are much, much longer. So a great example is starch. So starch is, again, it's just glucose, but hundreds of glucose units stuck together. And that's why you'll see these being called complex carbohydrates, because they're so long, it takes quite a long time for your body to use them, which is why you'll often see this referred to as being slow release energy. The other aspect of that is there's other things in bread, there's fiber and things that stop your body breaking that down so quickly. So that's what the polysaccharide is. It's just a load of sugar units stuck together. And there are a whole host of different polysaccharides. A lot of them are from plants. Bacteria make some polysaccharides. Some of them are obviously made from animals, or humans produce polysaccharides as well. One that you've probably come across previously is cellulose. So cellulose is essentially exactly the same as starch, except if you can see those oxygen bonds in between each sugar ring. In starch, both of them go down, or all of them go down. In cellulose, alternating, one goes down and the next one goes up. That changes the entire structure or conformation of that molecule. And what it means is that cellulose can't be broken down by enzymes in the human body, which is why it acts as a fiber, whereas starch can. So actually tiny, tiny changes in these chemical structures lead to really quite big changes in the function of these molecules. I've put a couple of examples here. So Gelan is made by bacteria. That's 4 different sugars in a particular orientation. Dextran sulfate is produced mainly in, well, it's produced in mammals, but mainly by bacteria. Methyl cellulose is essentially a modification. So again, you can take some of these polysaccharides, you can chemically modify them to give them different properties. But the ones I really want to draw your attention to are alginate, and in particular carrageenan, which are actually produced by seaweed. So both of these are produced by seaweed. You can eat all of these, and in fact, you'll find a lot of these in food, and they essentially act as fiber. So none of these in the human body can be digested. We don't have the enzymes to digest these, which is why they go straight through you and act as fiber. So why do we want to use polysaccharides as therapies? What are the advantages? Why are we interested in these molecules, sort of from a big picture perspective? So abundance, these are everywhere. Plants, bacteria, fungi. There's loads of different types of polysaccharides, and they're not difficult to get your hands on. Compared to traditional drugs, they are very low cost. Again, these are primarily used in the food industry, in cosmetics, as bulkers a lot of the time. So most interest in polysaccharides until now has been for their material properties, which we'll talk about at the end of this seminar. But they are very much cheap as chips. We're talking hundreds, thousands, 10,000 times as much for the same grammage of a therapeutic, even more for a biologic than we are for polysaccharides. Stability is an interesting one. So something we thought about is a lot of drugs, as I think a great example is from the COVID pandemic, when they were trying to roll out all these vaccines that had to be kept at -80. And that was a massive barrier, not only to deploying these in the UK, but trying to deploy them in places that don't have that infrastructure, that don't have that cold chain. Actually, stability becomes quite a big point. The last one actually is a long history of use. So as I mentioned, these have been used in the food industry, pharmaceuticals, cosmetics for a very, very long time. We know they're safe. We know what they do in the body, which makes turning these into therapies a lot quicker. If you have a molecule that's never existed before, you have to go through a lot of toxicology studies, lots and lots of animal modeling before it even gets to a human. Could it cause cancer? Could it cause genetic issues? Could it cause inflammation? All these types of things that with molecules that we know and have used for decades now, we don't have to deal with. And all of that really comes together if we're thinking about designing a therapy that people with EB might be using every day, maybe even multiple times a day. That's just not feasible with something that has to stay, in the freezer, costs £1,000 a time, all that kind of thing. So that's why that's where our interest in polysaccharides therapies really comes from. What do they do? Why we're interested in them? So main thing that they do and are used for in all these industries is structuring water. What I mean by that is they act as thickeners. So again, sorry to take it back to COVID. Again, I'm sure we'd all rather forget about it. But in alcohol hand gels, essentially to make them thick, so you could rub them over your hands, these have polysaccharides in there to provide that thickening effect. They also act as gelling agents. So most traditional jellies that you might have made use gelatin, which is actually denatured collagen. So that's using a protein. But a lot of vegetarian or vegan-friendly jellies, or a lot of the jellies that you find now, are made with polysaccharides. If any of you have ever made jelly, hopefully you remember that you use a very small amount of either powder or a small jelly cube added to a very large amount of water, probably from the kettle, to create quite a large amount of jelly. So actually that's what these molecules do when I talk about structuring water. Having these 1 part in 100 or normally less, so less than 1% polysaccharide, you can completely change the material properties of something that's mainly water. As an aside, if anyone's ever wondered why, when they hear our bodies are made of 70% water, how can that be? Because I don't feel myself sloshing around. This is why. Tiny, tiny amounts of long polymer molecules like polysaccharides can create a lot of structure out of water. So how does this relate to EB? Why we're interested in this for EB? So the first thing I want to talk about is how we can use these as lubricants. So this is trying to hit that first aim of stopping the injury from happening in the first place. So if we use something that acts as a lubricant, then essentially we can essentially use that to soak up those friction forces that perhaps occur between the teeth and the buccal epithelium or the toothbrush and that inside of the mouth, and actually stop that initial breakdown from happening in the first place. I'll come back to what the specific bars mean, but essentially you can see that on each of these, coefficient of friction is how much friction is caused between two plates when we put this stuff in between, and the lower that number, the better a lubricant our formulation is. So I think particularly if you look at the green bar, you can see that the green bar has almost a 5-fold lower coefficient of friction. And for chewing and brushing teeth, we're looking at around that 10 to 100 millimeter a second mark. So more towards the right-hand side of that first graph. So that's the first thing we can do, is we can formulate these to create lubricants, which might actually help through daily use, stopping some of that epithelial damage from happening. The next thing is stopping that collagen from forming. So if you remember, collagen is what the extracellular matrix is made out of. That's the cement that happens between the bricks. And that's essentially what too much is laid down during scarring. So normally, if we're in a situation where we want to stop scarring, we want to stop collagen being laid down. But what do I mean by laid down? So actually, collagen is a very complicated structure. So it's also a polymer, so it's a very long chain molecule, but instead of being a polysaccharide, it's a protein. What these do is they wrap into a triple helix. Then those triple helixes form in quite this complex manner into a fibril. And then the fibrils all bind together into a fiber. So it's not that dissimilar if you imagine textiles. If you think about a rope, you've got single fibers and then they all get twisted together. And then those twists get twisted together until you've gone from single molecules all the way up to tissues. So in the context of scarring, we actually want to try and interrupt this process if we can. So if you look at that top left figure, what we're showing here is in the absence of anything else, we just take, in this experiment, collagen. And this is a specific type of collagen where it acts as a liquid. And then as we warm it up, it starts that process of forming fibrils. And you can see in that top left figure, after about 20 minutes, half an hour, it started to form those fibrils. What we can see in the other graphs, so all these abbreviations are different types of polysaccharide. All of them, except for methylcellulose, at a certain concentration will slow that process. And then as we increase the concentration, it will stop it entirely. So you can see in B, C, D, and E, at those higher concentrations, there's just a flat line on the bottom, because after 6 hours, we left this for, no collagen fibril formation happens. So on a chemical level, some of these molecules can actually stop collagen fibril formation. Why? Well, what's interesting is that methylcellulose doesn't show this effect. And the way that methylcellulose is different to all the others is that it's neutrally charged. So all the other polysaccharides have negative charges on them. And what we think that does is it repels the collagen away from it and stops it from forming those fibrils. Whereas because methylcellulose doesn't have a charge on it, it doesn't stop those interactions from happening. So that's the second thing. We can hopefully limit how much damage is happening. We can also limit how much the collagen forms. And if we take this back to our brick wall analogy, this is kind of what we're getting at. So the polysaccharides there in yellow essentially get into that cement, almost the cement hardening process, if you like. It's very similar to the setting cement, and it can cause that cement to set slower. So again, that can limit the scarring formation that we see. Before we get into the biological bit, I just wanted to remind you of this process. So fibroblast is a skin cell. TGF-beta is that body that is that that signal that the body sends to the injury site that then triggers the fibroblast to turn into a myofibroblast. And what they do is they use alpha SMA to contract the wound, and they churn out loads of collagen, which is that cement. So key points to come into that this next section: TGF-beta, bad; alpha SMA, bad; collagen, bad. And we can essentially mimic this in a dish. So we can take normal fibroblast cells, we can culture them on a dish. So that's the picture on your left. And you can see, hopefully they look a little bit like my cartoon. They're sort of these crescent-shaped cells, and there's almost no alpha SMA present in these cells. When we add TGF-beta to this culture, basically the process that I just described happens. So some of these fibroblasts receive that signal, they differentiate and they turn into these myofibroblast cells. So a little bit more collagen perhaps, but the main thing you can see on the right are these green alpha smooth muscle actin, alpha SMA fibrils. And you can actually see in the image that these cross over, and you can see how these might start to contract the wound. So what we did is we essentially took this model and we introduced some of these different polysaccharides into the mix. And essentially, hopefully you can see, so NC is negative control, no TGF-beta, happy fibroblasts. PC is TGF-beta with nothing else, myofibroblast formation. And then you can see that some of these different polysaccharides have started to lower some of those signals. So you can see it in this image, the IC image, third along from the top. You have a lot less of that green signal from going on. And then if we quantify that on the bottom, how much of that there is, you can see that with the red arrows, a lot of those signals are coming down. So what we can see here is that this iota-carrageenan, which was that seaweed polysaccharide we talked about, actually dampens a lot of these signals. We can look at the same thing, but instead of looking at the proteins themselves, we can look at the genetic signal for those proteins. So for a cell to produce a protein, it has to send a signal from the nucleus out into the rest of the cell saying, we need to make this protein and this is how you do it. I really didn't mean to talk about COVID as much as I seem to be, but some of you will be familiar with PCR tests, and this is PCR. So essentially what we're doing is we take the cells, we extract the RNA, which is the signal for that formation, and then we amplify to see what signals are there, and then we can quantify them. So again, what you can see here is that some of those polysaccharides, particularly iota-carrageenan, are knocking that signal to produce collagen and alpha-racemate all the way down, essentially to normal. So that's really encouraging. You can see that it does this in a concentration-dependent manner, which from a scientific point of view is really important to understand how this might work. So essentially, the more of this carrageenan, this polysaccharide we put in, the better it damps that signal. And what that suggests to us is that what's happening is the carrageenan, the polysaccharide, is interacting directly with the TGF-beta. And we can see this in some of these direct measurements. So this kind of stuff is what I'm working on right now, is trying to understand the interaction between the iota-carrageenan, the polysaccharide, and that growth factor that causes the cells to turn into scarring cells. So what you can see on that figure on the left is that essentially that's the size of stuff in solution. So what we've done is we've put the polysaccharide into water, we've put the protein into water, and then we put them both together into water. And what we can see is when we put them in both together, we can see much larger particles. And what that's suggesting is that, yeah, the two things are coming together and forming sort of a big aggregate. Further evidence, this is data I collected last week with a colleague at Aston University, again, showing similar things. So the size is getting bigger, we're getting more spikes, which means there's more aggregates. And the figure on the right essentially shows a change in conformation of the protein, which suggests that indeed the polysaccharide is interacting directly with it and changing the shape of it in solution. So this is all, well, for me, certainly really exciting stuff. So hopefully I've convinced you at this point that we have 3 different strategies, all of which use the same polysaccharide and not much else, that we can use it as a lubricant. We can use it to stop those biological cues that halt the TGF-beta from acting, and we can stop it from physically causing scar formation. So these molecules seem really promising, but then how do we turn this into a product? How do we turn this into something that people with EB can use to hopefully try and slow or stop some of these symptoms? So what we wanted to do is design an oral spray. Now, the reason we wanted to design a spray, one is sprays are great for reaching hard to reach anatomical areas. So particularly up the nose is a great example. Nasal sprays have been around for a very long time, but also the inside of the mouth, especially if you've got less ability to open your mouth. Actually, spray delivery can be quite effective. And we can apply an even layer of material onto uneven surfaces, which again, the inside of the mouth has loads of. So this sounds great. Let's formulate this into a spray. The problem is that polysaccharides generally, and carrageenan in particular, sprays horribly. Well, it doesn't spray at all, really. If anything, this video makes it look quite good. But actually what it really does is it forms a jet. So it doesn't break up, it just shoots in a straight line and you don't get any breakup. So what we set out to do is try and formulate this material into something that we can spray while maintaining all those other properties that we were interested in. So we went back to the drawing board and thought, well, why doesn't this material spray well? And this is kind of why. So in just a solution of the polysaccharide, you can imagine if anyone's enjoys super noodles as much as I do, or you can think about spaghetti, all of these polymers are all entangled with all the other polymers, essentially into a big pile of spaghetti mess. What that means is there's nowhere in that material for it to break up easily. So if you think about spraying, what we want to do is break up that material in a very short space of time. And if you think about, if you've got your bowl of super noodles, if you try and pull it apart, it resists that quite strongly because all the noodles are intertwined with the other noodles. And essentially that stops that breakup from happening in the very short space of time we need it to. If you pulled your noodles slower, eventually they'd all start to rearrange and disentangle from each other. But because we try to do it fast in a spray, that doesn't happen. So what we wanted to do actually is then think about, why don't we go from a single phase material, so that entangled solution, that super noodle structure, into try and think about some more multi-phase systems. So systems where there's multiple things going on. So two of the ones that we've looked at so far are what we call a phase separated blend. So that's where we've got one type of polysaccharide, the carrageenan, that likes to interact with itself. A second type of polysaccharide, in this case gelan, which is that bacterially derived one, but they don't like to overlap with each other. What that means is that you get the same structuring of water that I talked about, those same material properties, but you almost get this checkerboard pattern where each type of polysaccharide is only hanging out with its friends and not in between. So there's very few overlaps between the groups. And you can see that dotted line I've drawn is essentially where we might get breakup during spraying. The second one is what we call a fluid gel. So as with a lot of these polysaccharides, you can induce gelation in them. In this case, it's by adding ions. So positively charged calcium, normally ions, negatively charged polymers. It encourages them to interact more and you get more gel type properties. As you can imagine, that still doesn't solve the problem. If you have a single phase, you've almost just made that network stronger. But what it means is if we turn that single jelly into lots of little micro jellies, you then can think about this path between the micro jellies for breakup. So in a similar way, we're maintaining a lot of those material properties and structuring that we want, but introducing little breaking points so that we can start thinking about spraying these materials. So here's some data just to show that this works. So there's carrageenan again on its own, jetting. Gelan actually sprays a little bit better because it's a little bit smaller and breaks apart from itself a bit better. But when you start combining these things and you get that checkerboard pattern, you actually get better spraying than either component on its own. Similarly with the fluid gel, I've sort of not gone into too much detail on how we did this, but you actually have to get the right ion into the right part of the polysaccharide in order to get this effect. But again, massive increase in spray area, sort of 20 times as much just from doing this kind of structuring. I quickly want to go back to the lubrication slide because that's what all these different bars mean. So that green bar that I was highlighting earlier, that's the one that's got that polysaccharide, the carrageenan, that's been structured into that fluid gel material using calcium, and you don't get the same effect with water. And if you have a much lower concentration of the polysaccharide, then you don't get that lubricating effect at all. So I didn't mention it earlier, but you can improve spraying by just having less polysaccharide in because there's less entanglement. But you then lose not only the lubricating properties, but also the other material properties that make these materials so good. So retention on the inside of the mouth, how long it stays there, if it's not thick, if you take all the polysaccharide out of it, it'll act like water and it'll only stay there for a couple of seconds maximum. Whereas if we try and create structured gel materials from the polysaccharides, then we can start to increase the material properties, get lubrication, and get all that stuff we want. So you can see that actually this is quite a marked effect on the spraying. So we go from that jetting behavior, and now we've got really good spraying behavior. So that's everything I want to talk about. Thank you so much for listening. Hopefully I've managed to convince you that polysaccharides are an exciting therapeutic potential, both in terms of what they can do scientifically, but also bigger picture, why they're quite an economical and feasible thing to turn into a therapeutic. Carrageenan in particular seems to show very strong activity at doing this, and what we'd really like to know now is why, which is what I'm studying at the moment. And that then we can turn these materials into formulated products that real people can use, hopefully to improve their quality of life, because ultimately that is the end goal of this research. So I'd like to again thank you all for coming and letting me speak. Thank you to Deborah for giving me the opportunity. And if anyone's got any questions, I'd be really happy to answer them. Thank you.
Thank you, Tom. That was just so interesting and clear. I think easy to understand what your what the aims are here and how it could benefit people.
Hopefully.
Yeah, hopefully. Yeah, I'm sure people are thinking, please do feel free to put any questions into the chat. I was just wondering about it in terms of, obviously you've done a lot of work developing a spray. I'm just thinking about cracked lips and dry lips and how your lips crack in the winter. You can get lip gloss, not that I'm a lip gloss person necessarily, but you can get lip gloss as a roll-on type delivery system. Have you considered any other delivery systems that might work?
Yeah, so I'll be honest, I mean, as great as sprays are, I'm partly interested in them from a scientific perspective, because I'm an engineer and I think it's useful for a load of different things. The other reason is that we sort of developed some of this in parallel with some other products, namely nasal sprays. So actually, some of these materials I talked about are actually very similar to antiviral nasal sprays that we as a lab developed during COVID. So that's part of the reason why I'm a bit obsessed with sprays. But you're absolutely right. There's no reason why you couldn't formulate these things into all sorts of other products. I mean, the major downside of spraying that some of you have probably thought about is that a lot of people with EB don't have great digital function. So actually operating a spray device isn't necessarily the easiest thing for a lot of people. So you could just use these as liquids. I mean, one of the things I mentioned with the spraying is the ability to create a thin layer of material. That's really important if you've got a million pound a kilo drug in there that you need to maximize the efficiency of. But if you've got something like this where it's the polysaccharide is the therapeutic, actually you can just use it as a liquid, almost use it as a mouthwash potentially, and use it that way. So yeah, there are a lot of other options that you could explore for this.
Thank you. Yeah, it's really just good to hear that everything's being considered, I think. And you've sort of come on to what I was going to ask about next by talking about what else could go in with the spray. Now, this might be a bit naive. I'm thinking about when you've got a sore throat and you eat a spoon of honey, which is sort of honey is presumably some sort of a polysaccharide or a sugary solution that sticks on your throat. People claim it has antiseptic properties. Would it be possible to include an antibiotic or antiseptic into one of these sprays that might help people with wounds in their mouths?
Yeah, absolutely. So honey is interesting. So honey actually it's not a polysaccharide. It's a very, very concentrated solution of monosaccharides and disaccharides. So which is why the concentration of sugar is so high. So I mentioned that with the polysaccharides, we've got less than 1% integrate that structure. I think honeys get well up into the 60s, 70s, 80s% sugar to create the same amount of structuring. Same with syrups and things. But yeah, there's absolutely no reason why you couldn't incorporate other things into these. As I said, they're 90%, well, not even 99.5% water. So anything water soluble would absolutely fit into these materials. I mean, the way honey works is a bit more complex. So if anyone's interested while we're here, the way honey is antimicrobial is there is an enzyme in honey called glucose oxidase. And what that does is it turns glucose and water into hydrogen peroxide and gluconic acid. And that's what makes it antimicrobial, which is why honey lasts on your shelf for a long, long time. That and because the sugar content is so high that bacteria can't survive because of the osmosis. The reason that's challenging, I know this because I have a friend who works in this exact industry, is that it can't contact too much water. Otherwise, the enzyme uses up all the sugar before it's where you want it. But other types of antiseptics, absolutely, there's no reason why you couldn't incorporate those into the same product.
That's really interesting. Thank you. And for the extra bonus fact about honey, that's always useful. So, I mean, I'm hoping that we'll have some questions from our attendees, but I think you might have stunned everyone into silence. But my next question is about this sounds really hopeful to me. I mean, we do fund work on gene therapy and all sorts of futuristic cutting edge technologies. This almost sounds like something that could be available soonish. Can you tell me whereabouts you are along the making it available to people timeline?
Yes, so actually Agnes in the chat has asked a similar question. It looks very similar to the Norazite spray that's online. It's the same stuff. So when I talked about that we developed this in almost conjunction with a nasal spray, that Gellan carrageenan mix I talked about, that's what Norazite essentially is. And we initially developed it as a antiviral nasal spray because carrageenan is, I don't want to say coincidentally, because I don't know if it's a coincidence or not, that it happens to be very antiviral and also very anti-scarring. But essentially, yeah, that's exactly what the same thing is. So the stuff I talked about in this talk is pretty much the same as the Norazite spray. I think it's now also an oral spray, but it was developed in the same lab.
Thank you. And I'm just apologizing to everyone who I said, please keep writing your questions in the chat, because we've been told the chat is not activated. So please do use the Q&A. Glad for some redundancy in the system. And I've got here a question. May I ask how long this mouth spray sticks on open areas? So how long it would last before rinsing or rubbing away?
Yeah, that's a great question, and one that we currently don't know the answer to. Essentially because it's very complicated. There's a lot of stuff going on in your mouth. So if you sprayed it into your mouth and then sat there with your mouth open, it would probably last quite a long time. But because your mouth is constantly producing saliva, that will start moving it away. It'll start diluting it away from where it is. Obviously, if you take into account things like chewing, things like toothbrushing, that will start to move it away as well. Similar types of materials. So we don't have the data for this in the mouth. So I'm afraid I can't answer your question directly. I'm really sorry. But to give you an indication of these similar types of materials that we've looked at in the eye, a typical water-based eye drop won't last more than 30 seconds in your eye typically. Whereas a gel-based eye drop will often last hours. So that's the kind of increase we can expect with some of these materials.
It just sounds so very promising. And I guess now my mind's full of all thoughts about where else it could be used. So eyes, mouth, throat. I don't know. I know a lot of people have issues with going to the toilet with EB. I don't even know if this would be something that would be considered. So essentially, it's a lubricating substance.
Yeah. So yes, I mean there are a couple of people working on EB at the university. I'm by no means the only one. Looking at a lubricating eye drop is currently in the pipeline. That one is happening. Again, it's currently a mouth spray. So we are thinking about different ways that these similar type of technologies can help. Again, the trick to move these from an idea into an actual product is trying to make some of these shortcuts I talked about. So the fact that these things have been used for a very long time makes it much easier. With the Norazite, the fact that we had it available as a nasal spray first, people already used it, and then we suggested actually this might cause some anti-scarring activity. I don't think there's any actual data for the anti-scarring activity of the Norazite itself, so I think I need to be careful of what claims I make. But again, if it's the same stuff repackaged, it's then much easier to move that into market. So
that's really good to hear. I think this is going to be an issue where people with EB struggle to find anything that's formally approved for their own use for their symptoms. So it's useful to know whether things could be repurposed.
absolutely.
There's a question here in the chat. Could the polysaccharides be used for encapsulating lipid nanoplastics and then apply topically on the EB skin? So for skin rather than,
nanoplastics and apply topical on EB skin. Yes, is the short answer. It could. So as I mentioned, I mean, most of the research around polysaccharides has been for formulation things, so material properties, particularly bulk material properties. So making things thicker, making things gel. What you can then do, and a lot of research people are, is combine that with smaller scale drug delivery systems. So as great as the material properties of these things are, if you've got for example, a water-soluble drug, you can't really control the release of that. You put it in the polysaccharide matrix, and then once it's where it is, you can't really stop it from moving because it's in something that's 99% water. So what people often do is they'll take that polysaccharide bulk material and put things like lipid nanoparticles in there. So you can encapsulate hydrophobic drugs, so drugs that wouldn't normally like to interact with water. And then you can then change the properties of those nanoparticles to start changing the release rate of those drugs. So that's something we're very interested in as well, is how you control the release rate of some of these therapeutics. So rather than having a massive spike and then tail until you take more, trying to keep it more level. So yeah, absolutely, there's scope for that.
So a lot of different possibilities to be explored.
Yeah, that's countless.
So where are you taking this next? What's your next step for this product?
I think something that's really important is to understand actually how these interactions are happening. We've essentially shown that it is happening, at least in a dish, but currently it's trying to work out why. So I think one of the things that's really important is to understand actually what that interaction between the polysaccharide and the protein is and how strong it is. So something I mentioned in my talk is that that the scarring process and the healing process are very, very tightly linked. So you can't just genetically knock out TGF-beta, for example, which would be perhaps the first thought of a biologist in the situation. So actually it's introducing a lot of nuance into that situation, trying to dampen those signals without removing them entirely. And then once we've kind of got a good idea of how this works, then moving up through the sort of technological readiness level. So looking at animal models, there are a couple of animal models of EB, but none in this country. Which, I mean, as I understand it, the reasons that the Home Office don't want animal models of EB is because it's such a horrible condition they don't want to expose animals to. But people have this condition, so that argument has never really made sense to me. But essentially trying to move it up through that way. And again, the other one that we've is happening, but is kind of more difficult to quantify, is that people with EB have been using the Norazite products, and as far as I understand it, do seem to see a benefit from that. So as you mentioned, using things off-label for EB, I think seems to work in some cases. And again, you've got to be very careful at the claims you make. And obviously, this evidence is anecdotal. So this is giving one person or a handful of people this thing and then asking their opinion afterwards. So you kind of get into the tricky bits with data versus a couple of people said it was great. But I mean, the couple of people saying it's great is A, very promising, but B, in itself, if it's helping even a handful of people, then that's great.
Yeah, I think that is wonderful. That's where we are at the moment, isn't it?
Yeah.
That is absolutely brilliant. I'm feeling that we're coming to the end of the questions from the chat, and
Okay.
if I take over on the screen sharing, I'll be able to share up the advert for the next one. But what I would really like to do first is just thank you so much for that presentation. It really was informative, full of hope, and full of promise for things that are coming down the pipeline.
No, thank you. So thank you very much for inviting me, and thank you everyone else for coming.